So welcome students. Today we are going to discuss about a very important cell component, that is the plasma membrane. In front of you on the screen is the plasma membrane, and if we have to describe its structure, then we always go to the proposal that was given by Singer and Nicholson, the two scientists who gave the fluid mosaic model, and we will describe that model here. So the structure of the plasma membrane, according to the fluid mosaic model, it has got two lipid bilayers. So you can see here this one, this upper sheath, this is the first layer, and then below this there is a second layer. So these are the two lipid bilayer, and now this lipid is called as a phospholipid. So the name of the lipid is phospholipid bilayer is present. So this and this are the two layers of lipid. Now in this lipid you can see some purple colored structure which are integrated into the membrane. Now this purple colored structure, this is the integral protein. This is called as an integral protein because it is completely embedded in between the two layers. Now apart from this, you can also see that some of the proteins, they are just present on any of the layer. It has not transversed completely, but it is present on either of the layer. So this kind of layer which is towards the surface will be called as a peripheral protein. So apart from this, there are certain glycoproteins which are also present on this membrane. So glycoprotein is nothing but it is a compound formed by the derivation of carbohydrate and proteins. So this was regarding the structure. In the structure, there is the phospholipid bilayer, and which has got two different kinds of protein, that is the peripheral protein and the integral protein, given by Singer and Nicholson, called as the fluid mosaic model. Now what is the function of this membrane? This membrane, it is obviously for protection because it is on the boundary of the cell, and many cell, especially animal cell, will have only the plasma membrane. So the primary function is protection. Second, it will provide some shape and rigidity to the cell. And third, it will also help in transport of material across the membrane. So this membrane is not extremely rigid. It will allow certain materials selectively to enter and to exit. Thus, it is also called as semi-permeable or selectively permeable. Selectively permeable membrane. Also, this membrane, it is fluid in nature. That means the particles, the phospholipid molecules here, they will be able to exchange places. It is not very rigid. It will allow the movement of the phospholipid in between the sheet. So it is called as the fluid. Students, in front of you on the screen is the nucleus. Nucleus in animal cell is present in the center of the cell, and in the plant cell it will be present in towards the periphery because of the presence of a large vacuole. Now when you look at the nucleus, it is said to be the controlling center, and this nucleus, it is covered with a nuclear membrane. This is the nuclear membrane. And then we find that on this membrane there are certain small pores. So these pores, these gaps or pores, they will help in exchange of materials into and outside the nucleus. So this is the nuclear pore. Inside we will find that it is filled with nucleoplasm. There is a fluid inside. That fluid is called as nucleoplasm. Now we will also find that there is a round dense structure which is called as a nucleolus. This is the nucleolus, and the function of the nucleolus is to produce ribosomes and RNAs. Inside, we also see large amount of thread-like structures. These thread-like structures, these are the chromatin materials. Chromatin materials, they will get converted into chromosome when the cell decides to divide. So these are the chromatin fibers. The chromatin fibers, they are the bearer of the hereditary material. Also, we know that they will get converted into chromosomes when the cell divide. So chromosomes or chromatin, both can be labeled here. But because these are thread-like, so it will be called as a chromatin. And when the chromatin fibers, they get condensed and folded into a rod-like structure, that time it will be called as a chromosome. So these are the bearers of hereditary material. And around this nucleus, we will find that starting from the nuclear membrane itself, we find that the extension of the endoplasmic reticulum takes place. Actually, endoplasmic reticulum is formed just by blobbing out of the membrane of this nuclear membrane itself. So let us discuss the next organelle as the endoplasmic reticulum. Students, this is an endoplasmic reticulum, as you can see, and this is the nucleus. This region is the nucleus, and you can see from the wall of the nuclear membrane itself has blabbed out and given rise to this flat tubular sac-like structure, and these are forming the network which is called as the endoplasmic reticulum. Here, each flattened sac-like structure, it is called as a cisternae. Now, this cisternae, they are connected to each other. We can see there are many layers of this cisternae and they are all connected to each other. Also, we can see that there are certain blue-colored dots, blue-colored things that are present on this membrane. And this blue colored dots, they are called as a ribosome. So this is the ribosome that is present on the cisternae here. Now you can see that this kind of cisternae like structure, it is also present in yellow color in this side after the blue colored ones. Now, is there any difference between both of them? Yes, there is some difference between them. The blue colored cisternae, the stack of cisternae here, these which are carrying the ribosomes, they will be coming under the rough endoplasmic reticulum. So they are called as rough endoplasmic reticulum because of the presence of the ribosomes, which give it the appearance of rough nature. Now, just after this cisternae, we see that there are certain other which may be a little tubular in shape, okay, flattened sac-like. These are also cisternae-like structure, and some may be little roundish, okay, and cylindrical. And these are showing sometimes some branching also. We can see here certain branchings are also seen here. Now, these where the ribosomes are absent, these kind of endoplasmic reticulum, they are called as smooth endoplasmic reticulum. Now, what is the function of rough endoplasmic reticulum? Rough endoplasmic reticulum's function is to synthesize protein. Because of the ribosome, it will synthesize proteins. Ribosomes are called as the protein factories. And then the smooth endoplasmic reticulum, because they do not have ribosomes, so they appear smooth, and their main function will be synthesis of lipids or fats. Lipids means fats. So all kinds of lipids, fats, steroids, and certain kind of sex hormones, the steroid-based sex hormones like the male and the female sex hormone, the testosterone and estrogen, they are produced from here. So hope students you have understood. Now just adjacent to this smooth endoplasmic reticulum, here in this area we will also find a Golgi apparatus. So let us discuss about that Golgi apparatus now. So students, we are going to discuss about the Golgi apparatus now. As you can see in the screen in front of you, the Golgi apparatus, this is the Golgi apparatus. The Golgi apparatus, it is composed of quite similar structure like the endoplasmic apparatus or the endoplasmic reticulum. Students, now in front of you in the screen is the Golgi apparatus. The Golgi apparatus, it has many components which are similar to the endoplasmic reticulum. Here also you can see that certain sac-like structure, it is stacked one upon the other. And this sac-like structures, these are called as the cisternae. Then there are certain tubular structures which will be present in the periphery of the cisternae. These are the tubules. And then you can see some round structures, spherical structures, which are blobbing out from these tubules or from the cisternae. And these are called as the vesicles. Now this side of the Golgi apparatus, it will be facing towards the nucleus, and it is called as a formative phase. This side of the Golgi apparatus, it is facing the plasma membrane or cell wall and is the maturation phase. So in the formative phase, some things will be formed, will start forming, and the things will come out is matured form from the other side. Now in the endoplasmic reticulum, we know the lipids and the proteins will be formed by the smooth endoplasmic reticulum and rough endoplasmic reticulum respectively. So all those proteins and lipids, now they will come and join this formative phase in the form of this round vesicles. We can see small vesicles which are coming out from the endoplasmic reticulum, and they will get joined to the cisternae. Now in this cisternae, those lipids and those proteins, they will be packaged and modified to form the final product. So when the final product has been formed through this by transporting inside the cistern, then they will come out later on as the vesicles. And some of these vesicles which will carry the digestive enzymes, only those vesicles which will carry the digestive enzymes, like this one, like here, okay, that is the hydrolytic enzyme, they will be called as the lysosome. Now certain vesicles, they will also be carrying those final products which are formed and will be acting like a storage organs. So to them we may call as vacuoles. We may also call them as vacuoles. So they are the storage organs that are present in the animal cells. That is the vacuoles. So the lysosome, students, you must remember the difference between vacuoles and lysosome. The lysosome are the one which will be carrying the digestive enzyme, and the vacuoles are the one which will be carrying the other final products of proteins and lipids and are the storage organs here. Now in this upper portion of the screen, you can see that how the lysosome works. Now here you can see lysosome has a membrane around it, and it is filled with the digestive enzymes. We can see that this is the plasma membrane. Okay, this boundary that is shown here, it is the plasma membrane. So this one is the plasma membrane. Now inside the plasma membrane, certain food particles, they enter. When they enter, this will form the food vacuole here, and this food vacuole, as it is entering more inside the cell, some of the lysosomes will find it, and the lysosomes, they will quickly release their digestive enzyme into this lysosome, and the food particles will be digested in this manner. And if there is any waste material produced, then the waste material will be again thrown back through the plasma membrane in the same manner. So this is how the lysosome works. Students, in front of you on the screen is a mitochondria. When you look at this mitochondria, you will find that there are two membranes. It is covered with two membranes. The first membrane, which appears very smooth, is the outer membrane of the mitochondria. It is the outer membrane and it is very smooth. Okay, let me write the name of the organelle, mitochondria. Sorry for the mistake. So this is the mitochondria, outer surface being smooth. Now look at the inner membrane. The inner membrane, it is producing large amount of folds. It has folded so much in order to increase the surface area of the organelle. So this inner surface is given into fold. So this is the inner membrane. And this inner membrane, the foldings that are formed, some of them are very thin, finger-like, deeply embedded inside. So these finger-like projections, they are called as the cristae. This is the cristae. Now on this cristae, we will find that they are not very smooth. There will be certain particles which are present on this cristae. Now these particles, they are called as oxysomes, and they will be responsible for making the ATP molecules or the energy currency of the cell. So oxysomes, or they are also called as F particles. So they have this two different names, oxysomes or F particles. And inside there is a blue color fluid that you can see. This fluid is called as a matrix. So this is the matrix. And then inside there are free floating ribosomes also present. So there are certain free floating ribosomes. Now, if they have ribosomes, they must be doing protein synthesis. So, for doing the protein synthesis, this kind of mitochondria, they also will have a circular DNA present in them. Mitochondria has got a circular single DNA. The DNA is circular. That means both the ends of the chromatin fiber, it is attached together. That's why we call it circular, and it is a single one. Now, because of the presence of this DNA and because of the presence of ribosome, now we know that it will produce its own protein. So this kind of a organelle which can produce its own protein, of course not everything it will prepare, but it will prepare many of the important ones. So it is also called as semi-autonomous organelle. Semi-autonomous organelle, because it does not depend upon the cell for every other protein. It can produce its own as well. So now what is the function of this? The oxysomes, we know they will produce ATP molecules, adenosine triphosphate molecules, and this adenosine triphosphate, it will give energy to any of the chemical reactions that will take place inside the cell. So that is the main function of the mitochondria. This structure that you can see on your screen is the plastid. Now the plastid is present only in the plant cell, and there are three different kinds of plastid. First one is the chloroplast, which has got green colored pigment, and because of the presence of chlorophyll. Then the second one is the chromoplast, which will have yellow orange colored pigments, and they are called as the chromoplast. And the third one is called as a leucoplast. They do not have any pigment, so they will appear transparent, and they will act as a storage organ for the cell. Now this plastid that you can see on your screen, it is a chloroplast because it is green in color. If you look at it carefully, structurally, it has got two membranes. So it has got two membranes here. So double membrane, outer and inner membrane, and both the membranes are very smooth. Double membranes. Now inside it is filled with a fluid which is called as a stroma. Stroma is the fluid which fills it up, and inside you can see that certain stacks, like coin stacks, are present. Now these coins, each one, they are called as a thylakoid, and the membrane which covers this thylakoid will be called as a thylakoid membrane. Now this stack of thylakoid, it will be given the name, this stack of thylakoid, it will be given the name as grana or granum. Granum is singular, and all the stacks together we will call it grana. So all the stacks together in the plural form we will call it grana. Now, where is the chlorophyll present? Chlorophyll will be present on the membrane of this thylakoid. So, on the thylakoid membrane, the chlorophyll pigment is present. Chlorophyll is just a molecule. Okay, so the chlorophyll pigment is present on the thylakoid membrane. Now, what is the function of a chloroplast or any of the plastid? The first function of the chloroplast, that it will absorb the sunlight and it will perform photosynthesis. And what is the function of the chromoplast? It will make fruits and different leaves very colorful, imparting it different colors, and even it gives color to the flowers and ripened fruit. Then leucoplast, it will act like a storage organ for storing extra food material inside the plant cell. So thank you everybody.